Welcome back to the deep dive. Today, we're taking a really close look at some critical sources.
That's right. Straight from the Advisory Committee on Immunization Practices, the ACIP deliberations.
And our goal here is to pull out the key intelligence, the stuff you, as a practicing clinician, really need to understand today's, well, pretty complex vaccine landscape.
Yeah, this isn't about the public noise. It's about the hard data, the methodology, those nuanced clinical trade-offs we all face.
Exactly. So think of this as a deep technical examination, really tailored for you, the MDs listening. We're pushing past those simple efficacy numbers you see everywhere.
We absolutely have to. We'll be dissecting methodological biases, trying to contextualize risk, using metrics like number needed to vaccinate, NNV, and ICER.
And critically, grappling with these emerging immune signals from the newer platforms, things that, frankly, demand we reassess how we approach informed consent.
Precisely. It's about patient counseling and maintaining trust in the face of evolving science.
Okay, so we've got a pretty challenging set of sources today. They really force us to balance that population health perspective with the individual patient risk right in front of you. We're going to break it down into three main areas. First, we'll start maybe smaller scale with the pediatric debate around MMRV.
Right, the febrile seizures issue. And how tricky it is counseling parents when there's a known avoidable adverse event, even if it's usually benign.
Then second, we're shifting gears more towards policy. We'll use the hepatitis B birth dose as a case study.
Yeah, looking at why targeted strategies sometimes fail historically and how gaps in our public health infrastructure can, well, force these universal policies.
Even if they create dilemmas at the individual level.
Yeah.
And we'll definitely hit on data quality issues there.
Oh, absolutely. Including that really insidious problem called healthy vaccine biases. It pops up again and again.
Okay, and third, and look, this is probably the longest and arguably the most critical section for your practice. Right now, we are dedicating serious time to the latest analysis of mRNA vaccine mechanisms.
This is crucial. We're talking biodistribution, persistence, what this IgG4 class switching actually means.
And connecting those dots to the accumulating evidence on chronic immune consequences. This is where we really get into the discussion about potential vaccine-induced autoimmunity and post-vaccine syndrome, stuff you need to know.
Exactly. It's essential for understanding the full picture today.
All right, let's kick off with a classic clinical headache, the MMRV vaccine. Proquad, right. Licensed way back in 2005.
Almost 2 decades ago, yeah. And the public health logic seemed pretty solid at the time.
Measles, mumps, rubella, varicella, 4-in-1 shot. Fewer needles, happier parents, better compliance. Seems like a win-win.
That was definitely the goal. Reduce needle sticks, especially in that age group where appointments can be stressful anyway.
And the immunogenicity data, it held up.
It did. The study showed it was non-inferior to giving the MMR and the varicella shots separately. So from an efficacy standpoint, it worked.
There's always a but.
There is. The convenience came with a trade-off, and this was known even before it was licensed. An increased risk of immediate reactogenicity, specifically fever and that measles-like rash.
Ah, okay. Tell us about that difference, because even a few percentage points for apparent weighing risks, that feels significant.
It absolutely does. The pre-licensure data showed fever in about 21.5% of the kids who got MMRV.
Okay.
Compare that to 14.9% for those getting the separate shots. So you're looking at a risk difference of 6.6%.
6.6%. Not tiny when you're talking about your own child.
Not at all.
Yeah.
And that higher fever risk translates directly into the main concern here, febrile seizures.
Right. And for any clinician, especially pediatricians or family docs, febrile seizures are they're incredibly anxiety-provoking for families, even though we know they're usually medically benign.
Exactly. We're talking about kids typically between 6 months and 5 years old. The peak incidence is right around 14 to 18 months, which lines up perfectly with that first dose schedule.
And while they're usually short, less than 15 minutes, they almost always mean a trip to urgent care or the ER. Panic, that
parental anxiety, that's the real ethical challenge with this vaccine. And the post-licensure studies, they confirmed the risk wasn't just theoretical.
So the data held up after it was rolled out.
Yes. Data from the Vaccine Safety Data Link, the VSD, confirmed it. Specifically, in the window of days 7 through 10 after that first dose of MMRV.
Okay.
The risk of a febrile seizure was significantly higher compared to getting the shots separately.
Do we have a number on that? Come, Shire.
Yeah, they quantified the attributable risk. So the extra seizures basically caused by choosing the combo shot over separate injections. It was cited as 4.3 additional seizures per 10,000 doses given.
Wow. Okay, 4.3 per 10,000. So roughly 4 times the baseline risk just for the convenience of one fewer needle stick. That's a tough sell to a worried parent.
It's a very hard number. And you have to think about the clinical burden of that trade-off. A seizure, even a simple febrile one, it often triggers a whole cascade.
A full workup, right? ER visit, maybe blood tests, sometimes even more, just to rule out something serious.
Exactly. And the ACIP discussion really acknowledged that cascade effect. One member pointed out, even if the seizure itself is medically benign, the aftermath isn't. The parental fear, the ER visit, sometimes unnecessary tests. Those are real costs.
I remember reading one comment, something like a 15-minute seizure feels like an eternity to the family.
Precisely. It's an enormously long time, was the quote. And that single event, even though temporary, even though neurological outcomes are generally fine, it can completely shatter a parent's trust, not just in that vaccine, but potentially in the whole vaccination schedule.
So the committee wasn't just looking at the physical risk to the child. They were factoring in the behavioral outcome, the potential for increased vaccine hesitancy down the line.
That's exactly right. It's about the impact on the parent's willingness to come back for the next shots. For that child, maybe for future siblings too. It affects overall coverage rates, way beyond just counting needles.
If the combo shot gives you basically the same immunity, but introduces this avoidable scare that makes parents skip future vaccines,
then the population-level benefit of convenience might actually be negated by that behavioral risk.
Yeah.
It's a complex calculation.
But then the data on the second dose was different, wasn't it?
It was, and that's really interesting. For kids getting their second dose, typically around age 4 to 6, the study showed no increased risk of febrile seizures with MMRV compared to separate shots.
Why the difference?
It makes biological sense. The general underlying risk of having a febrile seizure just naturally drops off quite significantly after age 5 or so.
Oh.
You've essentially aged out the most susceptible group.
So the timing is crucial. The risk is really concentrated in that younger first dose window.
Exactly. The peak susceptibility window matters most.
Which leads to the final recommendation, the one that was in place pre-vote at least. Seems like a very nuanced compromise.
It really is. The CDC effectively recommended using separate MMR and Varicella shots for that first dose between 12 and 15 months, specifically because of that higher seizure risk.
Mitigating the known avoidable risk in the most vulnerable group.
Right. But then MMRV was generally seen as acceptable, even preferred maybe, for the second dose later on around 4-6 years, or even for the first dose if the child was older, like over 48 months when they started.
So it's a perfect example of policy trying to manage not just disease risk, but also clinical anxiety and the downstream effects of a specific known adverse event.
It shows a pretty rigorous analysis, I think. The priorities shifted from just needle reduction convenience to actually maintaining long-term public confidence by minimizing that known risk during the peak susceptibility period.
A textbook case of balancing individual risk communication with broader public health goals.
Couldn't have said it better.
Okay, let's shift gears now. We're moving from a pediatric convenience issue to something more about systemic challenges, using the hepatitis B birth dose as our example.
Right, the universal birth dose policy. It's often presented as this essential public health safety net.
And from a population health view, why is that mandate considered so critical? Even if, for many individual babies born in the U.S., the immediate risk of exposure seems pretty low.
Well, the necessity is really rooted in two things: the severity of transmission risk and, frankly, historical failures in screening.
Okay, unpack that. Severity first.
Vertical transmission mother to child at birth is incredibly dangerous with HepB. Up to 90% of infants infected at birth will go on to develop chronic infection.
And chronic HepB means?
Decades later, liver cirrhosis, liver failure, liver cancer. It's a major long-term killer. And second, the U.S. actually carries a pretty significant burden of chronic infection already. About 70% of the chronic cases here are in foreign-born individuals, most of whom were infected at birth or in early childhood in their home countries.
So the universal birth dose acts like a buffer, a catch-all, especially because we aren't consistently screening all pregnant women. That's the system gap.
That is a huge part of it. The data presented during the ACIP deliberations really highlighted how the U.S. system is, well, different. We were one of only 3 countries out of 38 they looked at that doesn't have universal health care.
Which directly impacts consistent prenatal screening.
Directly. There's an estimated screening gap, Something like 14% of pregnant women in the U.S. are not screened for their Hep B status before delivery.
14% is a lot.
It's a huge number. And targeted strategies, like trying to only vaccinate babies of known positive moms, they just failed historically. Plus, for acute cases reported later, only about a quarter of people could even identify a risk factor, so the virus spread silently.
The universal birth dose basically compensates for those systemic failures in screening and follow-up.
It's designed to. It's a safety net for the system's imperfections.
This policy creates a very specific clinical dilemma right there in the delivery room in the first 24 hours of life. For the clinician on the ground, what's the immediate risk introduced by giving that vaccine so early?
And this is the crux of the debate. It's the reactogenicity. Pooled data from various studies shows adverse events within 24 hours of birth. Fever was reported in up to 5.6% of newborns.
Okay, 5.6%. And drowsiness, sleepiness?
That was reported much higher, up to 31.1%. But the fever, that 5.6%, is the one that triggers the big clinical cascade.
Because any fever in a neonate, an infant under 28 days old, is basically treated as sepsis until proven otherwise.
Exactly right. It's an automatic full sepsis workup. That means blood cultures, urine cultures, often a lumbar puncture, a spinal tap mandatory hospitalization for usually 48 hours, and starting empiric 5E antibiotics immediately.
Wow. Okay. So the clinician is potentially trading the small but real risk that the vaccine itself causes a temporary fever,
Which then mandates this invasive, expensive, and incredibly stressful workup for the family.
against the huge public health benefit of ensuring that infant is protected from potentially developing a lifelong, deadly chronic infection.
That's the trade-off in a nutshell.
So the debate then logically moves to, okay, if we know the mother is hepatitis B surface antigen negative, HBs ag negative, why not just delay the shot? Give it at the 1-month checkup instead. Avoid that day zero fever risk altogether.
Clinically, that sounds perfectly reasonable on the surface. You eliminate the chance of that vaccine-induced fever triggering the sepsis workup in a known low-risk situation. But public health officials raise the specter of loss to follow up. This is the killer argument against delaying.
How big a risk is that?
The data presented was pretty stark. It suggested that if an infant is not vaccinated at birth, the hazard ratio for failing to complete the entire Hep B vaccine series later is 3.
3 times more likely to not finish the series if you don't start it at birth.
Correct. So the argument from the public health side is, removing that very small risk of a sepsis workup isn't worth tripling the risk that the child never gets fully protected.
Especially since household exposure can happen later too.
Right. They cited data that even among children born to negative mothers, something like 7 to 11%, eventually test positive later in childhood, likely due to household or other close contact exposures. So for the policy folks, ensuring that first dose happens before discharge is paramount. Loss to follow-up is seen as the greater public health threat than the occasional fever workup.
Which brings us, again, to the quality of the data underpinning these decisions. The sources flagged a couple of really significant methodological concerns with the efficacy studies used. Conflicts of interest, for one.
Yeah, that was noted.
Yeah.
A review looked at the core efficacy studies supporting the universal policy. It found that 8 out of 17 key studies declared conflicts of interest.
What kind of conflict?
Primarily direct funding from pharmaceutical companies. Now look, a conflict of interest doesn't automatically mean that data is bad,
Sure.
but it absolutely means you need to apply extra scrutiny. And when you see that alongside other potential methodological weaknesses, it just heightens the need for caution in how you interpret the findings.
Okay. And the second big methodological flag was this thing called healthy vaccine bias. This seems really important for clinicians interpreting any observational vaccine data. How did it show up in the Hep B surveillance?
This is a fascinating and slightly terrifying example of selection bias in action. It came up in the adverse event surveillance data for preterm infants who received the Hep B vaccine.
Okay, what did they find?
Astonishingly, the data seemed to show that vaccinated preterm babies had a protective effect against developing bronchopulmonary dysplasia, or BPD.
Wait, BPD. That's the chronic lung disease premature babies get, often from needing ventilators, right?
Exactly. It's related to lung immaturity and injury from oxygen or ventilation.
So the vaccine, which protects against a liver virus, somehow appeared to prevent a chronic lung condition caused by prematurity.
Makes zero biological sense, doesn't it?
None whatsoever. That sounds like a massive red flag.
It absolutely is. And ACIP members jumped on this. They pointed out this finding strongly suggests it's a data artifact, not a real effect. It's classic healthy vaccine bias.
Explain how that works here. Why would vaccinated preemies look healthier in this way?
Think about which preterm infants actually get vaccinated according to schedule. It's the ones who are stable enough, strong enough, healthy enough to perhaps be discharged from the NICU, or at least stable enough that the clinical team feels comfortable giving a vaccine.
Okay, so they're already a healthier subset compared to the really fragile preemies who might still be critically ill.
Precisely. And crucially, they're also the infants whose parents are likely able to manage follow-up appointments, indicating potentially higher socioeconomic status, more engagement with the healthcare system overall.
Got it. So the group getting vaccinated isn't a random sample of all preterm infants. They are pre-selected for being healthier and having more engaged caregivers.
Exactly. So when you compare that group's outcomes, like lower rates of BPD, to the outcomes of the general preterm population, which includes the very sickest babies who never got vaccinated on schedule, the vaccine appears protective.
But it wasn't the vaccine conferring protection against BPD. It was the underlying health and stability of the babies who were selected to receive the vaccine.
That's the bias. The apparent efficacy wasn't real. It was an artifact of comparing fundamentally different groups. For clinicians looking at real-world evidence or observational studies on vaccine outcomes, this is a critical lesson. You have to constantly ask, could healthy user bias explain this finding? Because it often can, and it can create results that defy biology.
A vital reminder to be skeptical of observational data, especially when it looks too good to be true or biologically implausible.
Absolutely essential. Always question the methodology.
All right, let's transition now. We're moving into what might be the most consequential territory for everyone listening today, the novel mRNA platform.
Yeah, this is where the data gets really complex and, frankly, challenges a lot of the initial assumptions about how these vaccines work and their long-term effects.
We really need to analyze the data that questions that early narrative of, transient, localized, safe, and effective, and dive deep into the potential for chronic immune modulation and, yes, autoimmunity.
This information is absolutely critical for every informed consent discussion you have regarding these platforms, whether it's COVID vaccines or future mRNA therapeutics.
We have to move beyond thinking it's just about generating neutralizing antibodies and then it's all gone.
We do. The sources we reviewed point towards evidence of persistence, systemic distribution, and crucially long-term immune reprogramming that we as clinicians really need to grapple with. Hashtag D1, immune modulation, beyond neutralization.
Okay, let's start with maybe the most striking immunological finding, this IgG4 class switching phenomenon.
Right. Multiple studies now, particularly looking at people who received repeated doses of the Pfizer mRNA vaccine, BNT162b2.
Yeah.
They consistently show the development of unusually high levels of anti-spike antibodies belonging to the IgG4 subclass.
And why is that specific subclass, IgG4, raising eyebrows? It's not the typical antibody response we expect from a vaccine, is it?
Not at all for an acute infection or a typical vaccine response aiming for robust immunity. IgG4 is generally associated with immune tolerance or desensitization.
Tolerance, like what you see with allergy shots.
Exactly like that. It tends to develop after years of chronic high-dose antigen exposure. Think beekeepers getting stung constantly or patients undergoing allergen immunotherapy. It's fundamentally a non-inflammatory antibody isotype. It doesn't fix complement well. It doesn't trigger antibody-dependent cellular cytotoxicity effectively.
So seeing this rapid switch to high levels of IgG4 after just a few doses of an mRNA vaccine is unusual.
It's unprecedented, really, in this context, and it raises profound questions about what kind of immune reshaping is happening long-term.
So the concern is, instead of maintaining a strong, aggressive, neutralizing response, the kind you want to quickly clear a pathogen repeated dosing might be pushing the immune system towards a state of non-reactivity or tolerance, specifically to that spike antigen.
That's the core implication being discussed. It represents a significant shift in immune programming. This involves changes in underlying cytokine profiles, shifts in T-cell ratios, potentially in a very durable way.
And the clinical worry there is what? What could that tolerance lead to?
Well, there are a few concerns. One is whether this tolerance shift might impair the immune system's ability to mount effective responses against other antigens later on, maybe increasing vulnerability to future, perhaps different respiratory infections. And critically, for our discussion today, could this tolerance mechanism somehow paradoxically contribute to chronic inflammation or autoimmunity? If the immune system's normal surveillance and clearance mechanisms are being dampened or dysregulated specifically towards this widely expressed antigen, could that allow for pathology to develop? or even contribute to breaking self-tolerance down the line.
So the immune system is being told, ignore this thing, even while the thing might still be present or causing issues. That sounds potentially problematic.
It's an area demanding urgent research. We're seeing an immune signature normally associated with chronic tolerance developing rapidly after vaccination. We need to understand the long-term clinical consequences of that reprogramming. #D2. Biodistribution and persistence. A pharmacological concern.
Okay, let's talk about where this stuff actually goes in the body. The initial promise, or at least the public messaging, was that the mRNA and the resulting spike protein would be pretty short-lived, stay near the injection site, maybe the local lymph nodes, and then clear out quickly.
Right. That was the early narrative. Transient and localized.
But the preclinical data and now human data tells a very different story, doesn't it?
It really does. That initial assumption of confinement has been, well, systematically debunked by the evidence presented. The preclinical studies, often using reporter systems like luciferase mRNA, packaged in similar LNPs, they showed systemic distribution. The signal wasn't just in the muscle or lymph node. It was detected across multiple organs, liver, spleen, adrenal glands, ovaries, testes, and significantly the brain.
The brain. So the LNPs can cross the blood-brain barrier.
The evidence indicates they can. The lipid nanoparticles are essentially very effective systemic delivery vehicles. They are explicitly not confined to the injection site.
That completely changes the pharmacological risk assessment, doesn't it?
Yeah.
If the payload, the mRNA instructing cells to make spike protein, is getting delivered systemically,
then the potential for off-target effects, for unintended consequences in sensitive tissues far from the injection site, is vastly increased. It's no longer just a local immune event.
And this isn't just theoretical based on animal models. We have human data confirming this now.
We do. And this is perhaps even more concerning. The persistence data in humans is quite striking. Studies have detected both the vaccine mRNA itself and the spike protein it produces in various human tissues long after vaccination.
Which tissues? And for how long?
We're talking blood, lymph nodes, heart tissue biopsies, even brain tissue in some autopsy studies. And the duration is not days or weeks. We're seeing persistence for weeks, months. And in some documented cases, vaccine mRNA has been detected up to 706 days post-vaccination.
700? That's nearly 2 years.
Nearly 2 years. Let that sink in.
So we have a situation where the body is potentially being instructed to produce a foreign viral protein systemically in multiple organs, including the heart and brain, for months or even years.
That's what the data suggests is possible.
How does that connect to the concerns about autoimmunity and chronic illness for our listeners? This seems like a crucial mechanistic link.
It absolutely is. This persistence is arguably the linchpin connecting the vaccine platform to the potential for chronic disease. Think about it. If you have continuous production of a foreign antigen, spike protein, inside your own cells, in tissues where it doesn't belong, for a prolonged period,
Months, maybe years.
that dramatically increases the window of opportunity for immune dysregulation. It provides a constant stimulus. This is where mechanisms like molecular mimicry become much more plausible.
Explain molecular mimicry quickly.
It's where fragments of the foreign protein, the spike protein in this case, look very similar molecularly to fragments of our own human proteins. The immune system mounts an attack against the foreign spike, but because of the similarity, it mistakenly starts attacking the body's own tissues that contain those similar-looking proteins.
And the longer the foreign protein hangs around, the more chance there is for that mistaken identity to happen and take hold.
Exactly. Prolonged antigen presence increases the risk of breaking self-tolerance. The immune system gets locked into fighting this persistent internal invader, and that chronic fight can spill over into attacking self. This is thought to be a key potential mechanism behind some of the diverse and severe symptoms reported in post-vaccine syndrome, or PVS essentially, a vaccine-triggered autoimmune or autoinflammatory state driven by that persistent antigen. #D3. DNA impurities and off-target expression.
Okay, beyond the intended mRNA and spike protein, there are also manufacturing and quality control concerns raised. Let's touch on those. First, this issue of the modified mRNA itself leading to errors.
Yes, the use of pseudoridine, the synthetic nucleotide modification, used to stabilize the mRNA and maybe reduce innate immune recognition. It seems to have an unintended consequence. Studies showed it increases the rate of ribosomal frame shifting by up to 10-fold compared to unmodified mRNA.
Frame shifting, meaning the ribosome slips on the mRNA strand and starts reading the genetic code in the wrong frame, producing garbled proteins.
Precisely. It means your cells are potentially generating not just the intended spike protein, but also a whole array of unintended off-target proteins because the reading frame got messed up.
And do we know what these off-target proteins are or what they do?
We don't. But we know the body mounts an immune response to them because antibodies to these frame-shifted proteins have been detected in vaccinated individuals. So we're generating unknown proteins, eliciting an immune response to them, and we have basically zero data on their potential toxicity or long-term immunogenicity. It's an unknown variable introduced by the modification itself.
Another layer of complexity. And then there was the whole issue of DNA contamination in the vials.
Right. Concerns were raised about residual plasmid DNA left over from the manufacturing process, specifically pieces of the bacterial plasmids used to generate the mRNA, which might contain sequences like the SV40 promoter enhancer.
And the worry there is potential interaction with our own genome.
Well, the bigger issue flagged was how the regulators assessed the risk of this DNA. The thresholds for how much residual DNA was acceptable in each vaccine lot were apparently based on WHO and ICH guidelines for naked DNA.
Meaning just plain DNA floating around.
Exactly. And naked DNA doesn't get into cells very efficiently on its own, but that's not what we have here.
Here, the DNA is packaged inside the same lipid nanoparticle delivery system as the mRNA.
Precisely. The LNP acts like a delivery vehicle, Trojan horse. It's designed to efficiently carry its payload into cells. So any residual plasmid DNA isn't just floating outside, it's getting actively transported inside the cell, and potentially even into the nucleus, facilitated by the LNP.
So the safety standards applied were for the wrong scenario. They didn't account for the LNP delivery mechanism, potentially making that DNA contamination far more biologically active.
That was the core criticism. The risk calculation might have been fundamentally flawed because it ignored the delivery system's ability to shuttle that DNA inside cells, raising concerns about potential genomic integration or unintended gene activation that wouldn't apply to naked DNA. It's a critical oversight in the risk assessment process. #D4, cardiovascular risk and genetics, the myocarditis signal.
Let's turn to the heart now. We've all heard about the myocarditis risk, especially in young men. But the data presented suggests the problem might be much, much bigger than just the clinically obvious cases that land people in the hospital.
This is a really sobering finding, and something every clinician needs to be aware of. The focus has been on overt, symptomatic myocarditis pericarditis.
But what about subclinical damage?
What did the studies find?
2 significant studies were highlighted that did something clever. They tested individuals' troponin levels before vaccination and then again after vaccination.
Troponin being the key blood biomarker for heart muscle damage, indicating a heart attack or injury like myocarditis.
Exactly. And these studies found that between 2 to 3% of vaccinated individuals had elevated troponin levels after the shot, levels indicating some degree of heart muscle injury, even if they didn't necessarily have chest pain or other classic symptoms.
2 to 3%. Let's just pause on that.
Yeah.
If you apply that rate across the millions and millions of doses administered,
You are potentially talking about hundreds of thousands, maybe millions of individuals worldwide who experience some level of subclinical heart damage post-vaccination.
damage that's currently flying completely under the radar of our safety surveillance systems. Completely. Because systems like VA-EARS or VSD, they primarily capture diagnoses made when someone seeks medical care for symptoms. They look for ICD-10 codes for myocarditis, resulting from hospital admission or an ER visit. They are structurally blind to this potentially massive burden of silent subclinical cardiac injury.
And what are the potential long-term consequences of even mild subclinical myocardial damage? It's not benign, is it?
Absolutely not. Any damage to the heart muscle can lead to scarring, fibrosis. That can potentially reduce cardiac reserve over time, increase the risk of arrhythmias later in life, or make the heart more vulnerable to future stressors. We could be creating a huge cohort of people with undetected heart injury, the consequences of which might only become apparent years down the road.
And even the tragic cases of overt severe myocarditis leading to death shortly after vaccination. Are they being properly counted?
That was another major concern raised. Published autopsy reports were discussed, citing cases of death, say 3 or 4 days after a second Pfizer dose. The pathologists described findings that were utterly not typical to viral infection.
Meaning it didn't look like typical myocarditis caused by a virus.
Correct. They described findings suggestive of a different mechanism, perhaps a catecholamine-driven hypersensitivity reaction, a fulminant myocarditis, directly triggered by the vaccine's inflammatory response. But because these deaths might occur just outside an arbitrary surveillance window, or because the mechanism isn't the classic viral one, there's a real concern they aren't being accurately attributed as vaccine-related deaths in our official statistics.
Which further compromises the integrity of the safety data we rely on.
It absolutely does.
Now, tying this back to our theme of immune dysregulation and potential autoimmunity, there's emerging data on genetic susceptibility to this myocarditis, right?
Yes, this is fascinating and points towards personalized risk. Sequencing studies performed on individuals who developed myocarditis after vaccination have started identifying specific genetic variants.
In which genes?
Genes involved in cardiac function, inflammation, immune response, things like Titin, TTN, AGTR1, LRP8, even VKRC1 involved in coagulation pathways.
And what was the pattern they observed?
An intriguing trend emerged, suggesting that patients who carried a higher number of these risk variants in a homozygous state, meaning they had two copies of the risk version of the gene, tended to develop myocarditis symptoms significantly sooner after vaccination.
So, more risk genes meant a faster onset of heart inflammation post-vaccine.
That's the suggestion. It implies that certain individuals are genetically primed, biologically predisposed, to have a severe, rapid inflammatory or potentially autoimmune reaction targeting their heart tissue upon encountering the spike antigen or the associated inflammation.
Which moves us away from just being a random, rare event.
And towards potentially being a predictable, genetically determined susceptibility in a subset of the population. This raises profound questions about whether we could or should be screening for these genetic risk factors, although that's obviously not feasible currently on a mass scale. But it strongly suggests an underlying biological, potentially autoimmune-like mechanism in those susceptible individuals. #D5. Post-vaccine syndrome, PVS, and surveillance gaps.
Okay, let's try to synthesize some of this. We have evidence for persistent antigen, immune modulation like IgG4 switching, potential off-target effects, subclinical organ damage, and genetic susceptibility. How does this all relate to the cluster of symptoms many clinicians are now seeing, often referred to as post-vaccine syndrome or PVS?
PVS is essentially the clinical manifestation that seems to tie many of these mechanistic threads together. It's being described by clinicians worldwide as a complex, often debilitating, multi-system illness emerging after COVID vaccination in some individuals.
What kind of symptoms are we talking about?
They are incredibly diverse, often overlapping heavily with symptoms seen in long COVID, interestingly. Things like chronic, widespread pain, profound, disabling fatigue, cognitive dysfunction or brain fog, postural orthostatic tachycardia syndrome, POTS, and other forms of dysautonomia, development or worsening of autoimmune diseases like small fiber neuropathy, rheumatoid arthritis, lupus, and severe neurological issues.
It sounds like a systemic breakdown affecting multiple organ systems.
It really does. And the unifying hypothesis, linking back to the mechanisms we discussed, is often centered on immune dysregulation. The idea is that in susceptible individuals, the vaccine, perhaps due to that persistent spike protein, or the inflammatory nature of the LNP, or the specific type of immune response generated, triggers a chronic inflammatory or autoimmune cascade.
So PVS could be, in many cases, a form of vaccine-induced autoimmunity or a related chronic inflammatory state.
That's a leading hypothesis based on the symptoms and the mechanistic data we have. The persistent antigen could be driving it, molecular mimicry could be playing a role, the IgG4 switch might be involved in modulating the response. It's likely complex and may be different pathways in different people.
Yet, despite clinicians reporting these cases, the official public health surveillance systems seem almost entirely unable to capture or quantify this syndrome. Why the disconnect?
It's a fundamental flaw in how our current safety surveillance systems are designed. Systems like the Vaccine Safety Data Link, VSD, which are often held up as the gold standard for monitoring. They primarily work by looking for pre-specified health outcomes, using existing ICD-10 diagnostic codes within specific time windows post-vaccination.
And there's no ICD-10 code for post-vaccine syndrome.
Exactly. There's no single code for it. There isn't even a widely accepted specific code for post-vaccination dysautonomia or vaccine-induced small fiber neuropathy. So when a patient presents with, say, new onset, POTS, chronic fatigue and neuropathy 2 months after their vaccination.
The system might just record POTS or neuropathy, maybe fatigue.
Right. It captures the individual symptoms or resulting diagnoses, but there's no built-in mechanism to systematically and reliably link that constellation of potentially related issues back to the vaccination as the potential trigger, especially if it emerges weeks or months later. The complex, multi-system, often delayed onset nature of PVS means it just falls right through the cracks of our current surveillance architecture.
So we have mounting biological evidence pointing to mechanisms that could cause such a syndrome, persistence, immune changes. We have clinicians reporting the clinical syndrome itself, but we have no official way to count how often it's happening.
That's the deeply concerning reality. It leaves the burden entirely on individual clinicians to recognize the pattern, make the potential connection, and manage these incredibly complex patients, often without clear diagnostic pathways or treatment protocols, and certainly without official acknowledgement or support from the broader public health system.
Which brings us back to the need for clinical humility. For the MD listening, what's the takeaway here regarding PVS?
The takeaway is, you need to take these reports seriously. Acknowledge the biological plausibility based on the persistence and immune modulation data. Listen to your patients. Don't dismiss these complex symptom clusters just because there isn't a neat ICD-10 code or because official surveillance isn't capturing it. Ignoring these patterns because they don't fit our current boxes is, frankly, an ethical lapse. We need to be vigilant, document these cases thoroughly, and advocate for better surveillance and research to understand and address PVS.
Okay, given the sheer complexity from the nuanced MMRV risk-benefit balancing act to the systemic failures highlighted by the HepB data, and now these profound uncertainties and potential long-term immune consequences of the mRNA platform, let's talk about how public health policy actually measures efficacy and risk.
Right? Because the methods used have significant implications.
A lot of the recent vaccine effectiveness data, especially for COVID boosters, relies heavily on something called the test negative design, or TND. You see it used in the Vision network, the IVY network. What are the major limitations of TND that clinicians really need to understand when they see those VE numbers?
TND is an observational study design, not a randomized controlled trial. It compares the vaccination status among people who seek care for symptoms and test positive for the virus versus those who seek care for similar symptoms but test negative.
It sounds quicker and cheaper than an RCT.
It is. That's its appeal. But it is profoundly vulnerable to confounding variables, especially selection bias and healthy user bias, the same kinds of issues we saw with the HepB finding.
How so in this context?
Well, who seeks testing? When do they seek it? Are vaccinated people more likely to get tested for mild symptoms because they're generally more health conscious? Are unvaccinated people perhaps delaying testing until they are much sicker? These behavioral differences between vaccinated and unvaccinated groups can heavily skew the results.
And critically, TND isn't really suitable for measuring effectiveness against the most important outcomes, like hospitalization or death.
Correct. It's primarily designed to estimate VE against symptomatic infection or maybe medically attended illness. It's considered not appropriate for reliably assessing VE against severe outcomes like hospitalization or death because the biases become even more pronounced when looking at those rarer, more extreme endpoints.
And some critiques presented at the ACIP meetings were even stronger, suggesting TND could be so biased it might even show protection when there isn't any.
Absolutely. Concerns were raised that due to these biases, TND could potentially suggest vaccine effectiveness even if the vaccine was actually neutral, or maybe even if it was associated with a negative effect on, say, other non-COVID respiratory infections. The healthy vaccine effect can be that strong.
So the bottom line for the clinician seeing a VE estimate from a TND study is extreme caution.
Extreme caution. Recognize its observational data, prone to significant bias. It is not the same level of evidence as an RCT, the gold standard.
Which leads to the repeated calls we heard in the source materials for more RCTs.
Yes. Strong calls for randomized controlled trials for all new vaccines, especially new platforms, and also for studying concomitant administration, giving multiple vaccines at the same time. The reliance on real-world evidence needs to be tempered by a deep understanding of its inherent limitations.
And this need for methodological rigor connects directly to the confidence, or perhaps overconfidence, sometimes expressed in public health messaging.
It does. Committee members themselves noted that the frequent assertion of safe and effective, while perhaps well-intentioned, often lacked the backing of that gold standard RCT science, especially when it came to novel platforms, long-term effects, or use in specific populations like pregnant women, where high-quality data was often missing.
Which brings us right back to the heart of the clinical encounter, informed consent. If the data is this complex, if there are known unknowns and potential long-term risks related to immune changes and autoimmunity, what does genuine, transparent, informed consent look like now for you, the physician?
It has to be radically different from just saying it's safe and effective. A minority work group within ACPP actually proposed some specific language, which I think provides a powerful template for what comprehensive consent should include.
What did they suggest?
They argued consent forms should mandatorily acknowledge several key points based on the evidence presented to the committee. First, acknowledge that the evidence supporting protection against severe outcomes like hospitalization or long COVID is often of low quality and primarily short-term. No guarantees of long-lasting protection.
Okay. That's honest about the evidence limitations. What else?
Second, explicitly disclose that evidence exists suggesting repeated boosters may cause acquired immune changes, referencing the IgG4 finding, and that this carries a potential for increased vulnerability to future respiratory infections due to that immune shaping. This addresses the tolerance concern directly.
Wow, that's a significant disclosure.
It is. And third, the consent must include the known documented risks. Specifically, mention the documented deaths that have occurred from myocarditis, pericarditis, and explicitly state the risk of developing a chronic, debilitating post-vaccine syndrome, PVS, involving the kinds of symptoms we discussed.
That proposed language is incredibly different from the standard reassurances patients usually receive. It really shifts the focus to the uncertainties and known risks.
It puts the power back with the patient by giving them the unfiltered scientific context, the good and the bad, the knowns and the unknowns. That's the foundation of true shared decision-making.
It really underscores the ethical mandate for clinicians, doesn't it? Especially when you're advising intervention for healthy people, the standard for safety evidence and transparency has to be exceptionally high.
It absolutely must be. Given everything we now know or suspect about persistence, biodistribution, immune shaping, subclinical injury, PVS, and the potential links to autoimmunity, clinicians have an ethical obligation to be radically transparent about these risks and the gaps in our long-term safety knowledge. Anything less fails the principle of informed consent.
One final policy point that came up was the practical debate around access. Should COVID-19 vaccines require a prescription from a doctor or continue to be available directly through pharmacists?
Yeah, that was a really contentious discussion, pitting the ideal of thorough physician counseling against the reality of public health access.
The argument for requiring a prescription being it forces that conversation with a doctor, potentially enabling the kind of detailed, informed consent we just discussed.
Right. It could facilitate a deeper risk-benefit discussion. However, the counter-argument was massive access.
Requiring a doctor's visit, especially in the current U.S. healthcare system, can mean long waits, difficulty getting appointments, particularly for primary care.
Exactly. Potentially year-long waits for new patient appointments with internists were mentioned. It would disproportionately impact people in rural areas, those with lower socioeconomic status, people who rely heavily on pharmacies for convenient healthcare access.
And the data showed pharmacists have been absolutely crucial for rollout.
Hugely. Something like 67% of all adult COVID vaccine doses were administered in pharmacies. So putting up a prescription barrier would likely decimate uptake rates, undermining the primary prevention goal.
So the decision was ultimately to prioritize access.
Yes. The vote favored maintaining broad access through pharmacists, recognizing that while the ideal counseling scenario might be with a physician, the practical reality is that restricting access would likely do more harm from a public health perspective by reducing vaccination rates significantly.
Which means the burden remains on the entire clinical community, physicians, pharmacists, nurses, to ensure that the information provided, the educational materials, the brief conversations that do happen are as rigorous and transparent as possible, regardless of the setting. That's the challenge. We need high-quality information and true informed consent, delivered through a system designed for high-throughput access. It's a difficult balance. #TagVax conclusion, the clinical mandate.
Okay, let's try to pull all of this together. We've covered a lot of ground. From the MMRV calculations that had to weigh seizure risk against parental anxiety,
to the systemic failures and data biases revealed by the hepatitis B case study.
And then this deep, frankly unsettling dive into the mRNA platform's pharmacology, persistence, and the profound questions about long-term immune effects and potential links to autoimmunity. It really demonstrates that the job of being a physician today, navigating vaccines, is just radically different, isn't it? It's not just about looking up efficacy numbers anymore.
Not at all. It's about synthesizing incredibly complex data, understanding methodological pitfalls, calculating nuanced risks, and confronting these significant uncertainties, especially around long-term immune consequences like IgG4 switching, persistence, and PVS.
So if there's one core takeaway for the MDs, the clinicians listening today, what is it? I think it has to be the need for radical clinical humility and absolute transparency. Given the evidence we've reviewed, the plausibility in reports of PVS, the reality of widespread biodistribution and persistence, the implications of IgG4 immune shaping, the clear methodological biases in much of the observational data,
Yeah.
we are ethically mandated to use shared decision-making in its truest sense. That means openly acknowledging the risks, the benefits, and the significant gaps in our long-term safety data, especially when advising otherwise healthy patients, and particularly when dealing with these novel platforms where we now know chronic immune shaping is occurring.
Yeah.
We just don't have all the answers yet, and we have to be honest about that.
And this leads us to a final thought, something maybe a bit provocative to leave our listeners with. It ties back directly to this idea of chronic disease and immune function.
Okay.
We've seen data presented right at ACIP suggesting that the protection you get from an updated COVID booster, for instance, is often moderate at best, and critically, it's short-term. Protection might wane significantly within just a few months.
That seems consistent with what we're observing, yes. Transient protection against infection.
Yet contrast that with the signals for immune changes. The persistence of the spike protein, potentially for months or years, the genetic predispositions influencing adverse events, the IgG4 class switching. These signals appear potentially long-term, maybe even chronic or permanent immune shifts.
That's a stark contrast. Short-term protection, long-term immune alteration.
Exactly. So here's the final question, the challenge really for everyone practicing medicine today. What is the actual clinical significance, the long-term wisdom of repeatedly generating a persistent antigen exposure and driving non-neutralizing tolerance-associated immune responses like IgG4, all directed against an ephemeral, constantly mutating seasonal virus? And how does that strategy creating chronic immune changes for short-term benefits inform how we should think about and perhaps manage the seemingly ever-increasing burden of complex chronic inflammatory conditions and potential autoimmune diseases that you are seeing in your patients every single day?
That is the multi-billion-dollar question, isn't it? How do these repeated interventions shape long-term population health and chronic disease trajectories?
It's the question you, the practitioners on the front lines, are going to have to grapple with and ultimately help answer in the coming years. Something to think about.